So, good morning, one and all present here for the guest lecture, which is based on the theme Indians Speaking from Abroad on the occasion of 124th birth anniversary of founder president of Sri Shivaji Education Society Amravati, Dr. Panjabrao alias Arvind Deshmukh, the Science College, Congress Nagar, Nagpur, in collaboration with Microbiologists Society India, organizing a guest lecture series which is based on the theme Indians Speaking from Abroad. So, on behalf of the organizing committee's Honorable Principal Professor Mahendra Dhore Sir, Science College, Congress Nagar, Nagpur, Honorable President, Microbiologists Society India, Dr. A.M. Deshmukh Sir, Honorable Coordinator, National Coordinator of Microbiologists Society India, Dr. S.D. Patankar Sir, and on behalf of the organizing committee, I welcome the eminent guest speaker of today's guest lecture, Dr. Trupti Gokhale ma'am. Welcome you and welcome all the participants of today's lecture. So the topic of today's lecture, which will be delivered by Dr. Trupti Gokhale ma'am, is novel biodegradable packaging material embedded with metal nanoparticles. So before us to start the lecture, I must take the opportunity to introduce our eminent guest speaker of today's guest lecture. Dr. Trupti Gokhale ma'am has completed her PhD in 2002 from Department of Microbiology, Goa University, Goa. Her post-graduation from Mumbai and her undergraduation is also from Mumbai in 1995. In the awards and scholarship, which she is the recipient of the Best Paper Award at the 3rd Global Sustainable Biotech Congress in 2014 at Jalgaon, India. Received the Best Paper Award for Technology Transfer at the Intech 2011 conference at Bratislava, Slovak Republic in 2011. Independent Research Associateship from Department of Biotechnology from DBT in 2003 to 2006. Scholarship from the Ocean Science and Technology Cell, Government of India for the year 2001-2002. Goa University Studentship for the year 2000 to 2001. Then scholarship from the Department of Ocean Development in 1997 to 2000. Mumbai University Student Scholarship in 1997. She is also nominated as an expert in the field of mangroves by the Mangrove Research Organization of National Institute of Oceanography, India. She has carried out some major research projects and received the grant from BITS Pilani, Dubai campus for the project entitled Optimization of Selected High-Yield Grass Species for Biomass and Bioenergy Applications using saline and recycled wastewater in 2017 to 2020. She has also received grant from Expo Life under the University of Innovation grant in 2018, co-principal investigator for the Red Mud Remediation Project by Hindalco Industry. She is also the advisory board member of many professional bodies like International Conference on Microbes for Better Tomorrow, as well as International Conference on Nanotechnology for Human Welfare. She is also the advisory board member for International Conference on Applied Nanotechnology and Nanosciences, as well as the International Conference on Microbial Technology for Better Tomorrow. Then International Advisory Board for International Conference on Biotechnology for Better Tomorrow in 2013. She has many contributions as a convener or as an organizing committee member of various national and international conferences. She has the teaching experience at undergraduate and postgraduate level from 2009 onwards at Department of Biotechnology, BITS Pilani, Dubai campus. And before that, she has a teaching experience at Department of Microbiology and Biotechnology at Wilson College, Mumbai, as well as the University of Pune, India. She has supervised many research students under her supervision. She has also carried out some invited talks, and she has invited for the talks on different conferences as well as the seminars and symposium. She has published a book chapter which is entitled Applications of Metal Nanoparticles in Agriculture in Shivaji Power. Then publications in international and national journals. She has many national and international journals published to her credit, as well as she has participated and presented the research papers in many national and international conferences. Apart from this, she has the postdoctoral work on two aspects: studying the membrane protein interaction and voids in the membrane as an effect of osmotic pressure on the membranes, as well as to exhibit the presence of an alternate oxidase respiratory pathway in eukaryotes to combat the osmotic stress and help in growth and accumulation of secondary metabolites. She has her research interest specifically in the metal nanoparticles synthesis as well as development of biosensors using the LUX genes, as well as development of bioplastics from bacteria. She has also her research interest in the development of biofertilizer using the native rhizobial cultures from desert soils, bioremediation of oil spills using halophilic bacteria capable of degrading recalcitrant hydrocarbons. The effects of microwaves on bacterial culture are being studied with respect to the damage caused by microwaves to the DNA and the proteins, as well as she has the research interest in effect of osmotic pressure on biological membranes. So thank you very much, ma'am, for accepting our invitation, for delivering your precious talk on your topic. It would be really grateful to us. So on behalf of the organizing committee, I take this opportunity to invite you, to request you to deliver your precious talk. And before that, please give some idea about your journey from India to abroad. Please, ma'am.
Thank you so much, Dr. Pranita. I think you summarized my entire career as well as my life in microbiology. It was pretty, you just took me down the lane of all my sweet memories through this entire process. Okay, so just to recap on my journey, I started with my microbiology career as a BSc student in Wilson College. Wilson College has been very close to me because I have throughout been a student of the John Wilson Education Society. My schooling was also under the same society in St. Columba School. I did my under, that is junior college, as well as my BSc in Wilson College. I also stood, I was one of the top rankers in during my BSc, and that's why I got a college of my choice for MSc in Mumbai University. So definitely I would opt for Wilson College. There was no compromise on that. So I'm like being a very dedicated student of John Wilson Education Society, yeah, starting from my school right up to my post-graduation. Unfortunately, PhD options then were not available with Wilson College, and that's why I had to move out. If I had a guide there, I think I would have even completed my PhD in Wilson College. So yeah, I got a good opportunity in Goa. I did my PhD with Dr. Mahendra under the DoD scholarship, that was Department of Ocean Development. And that's when I was introduced to the field of mangroves. I did initially work on studying the bioflora of mangroves, and then went on to select my PhD topic, which revolved around biodegradable plastics. So that is when I actually moved from microbiology into little interdisciplinary work in microbiology. So looking mostly at applications of microorganisms for a sustained environment. So my PhD topic did involve synthesis of biodegradable polymers. So I worked on polyhydroxyalkanoids from bacteria, and tried to study their mechanical properties, etc. Tried to develop methods that would make easy recovery of these from the organisms, because they're intracellular. So that's when I connected outside microbiology and ventured into polymer science. So a lot of work happened in polymer science. Like any other PhD student, I was eager to complete my PhD and move on. So because of course I was in Goa, I was staying in the hostel. That is one major, a thing that always remains at the back of your mind. You want to be back home. And I then moved to Pune, where my husband was. We got married while my PhD was on. So in Pune, I joined Pune University, Department of Biotechnology, under the guidance or supervision of Dr. Sitaramam. That is a lab which gave me more exposure to the interdisciplinary ideas. So that's where I got a little into biophysics, where we studied and focused mainly on the effect of osmotic pressure on membranes. So we tried to establish the alternate oxidase pathway in 3 different systems. We tried to look that in yeast, Dunaliella, and rice plants. So that is where I, again, because of yeast and Dunaliella, unicellular, that is where the microbiology part remained. But actually, I moved out from prokaryotes into eukaryotes to work over there. It was not so easy, but yes, it was quite challenging and nice work that we did. That is when I also got the IIC Bangalore DBT scholarship. Then I just happened to visit Wilson College once again during one of my vacations, and I was trying to look for some job in Mumbai because my husband moved to Mumbai. So I kind of like just followed him through my life. But yeah, it worked. So I just went to meet my teachers to say hi, hello, and they said, "Why are you looking for a job outside when we want a biotechnology person over here? We are starting the program in biotechnology." So it was nice for me. It was like a red carpet welcome. I just went to meet them, and I returned home with an appointment letter. So it was a really nice welcome. And that's how I started teaching in Wilson College. From biotechnology, I helped them establish the biotech lab, the entire biotech program. And meanwhile, I also taught microbiology, and I was a lecturer in microbiology. And when all this was looking very nice and good, my husband moved to Dubai. So I was very reluctant to move to Dubai because, Wilson College was a 5 minutes walk from my home. So you can imagine in Mumbai, somebody gets a job at a 5 minutes walking distance, would never want to leave that kind of a job and a UGC position to come to Dubai where nothing was known. I had no career options over here. So I kind of pulled down for 2 years, and then of course I had to move because definitely more than career, I did value my family. And I just, as I said, like always, I followed him. I left my job in Mumbai and I came to Dubai. I did struggle for a year and a half where I was absolutely without a job. But I kind of focused on my daughter. So I used that time for her upbringing. And then I got into BITS Pilani. So from 2009, I'm with BITS Pilani. So I completed 13 years over here in the Department of Biotechnology. And I guess all the experience that I gathered through is what I'm using right now in BITS Pilani. So here, of course, microbiology always remains. That doesn't go out. And I feel a person who's trained in microbiology can work in any field. Yeah, we are one set of extremely versatile, I'll say in skill wise, we are extremely versatile, and we will not see such skills coming up from other fields. But anybody who's a microbiologist gets very good hand skills. The techniques that we learn do not limit ourselves to microbiology. Even the analytical thinking is much beyond. And that is what I keep telling the students. And when I look for new recruits, if I see anybody who's done training in microbiology, of course, my bias always goes in that direction. Because I feel amongst the BSc lot or the MSc lot, they are the ones who are more skilled than any other field. Because of its versatility and a lot of diverse techniques that we learn. So yeah, over here, after I moved, I initially started working on biofertilizers, trying to look for organisms in the sand, absolutely arid sand. But then we first focused on the rhizosphere area around the leguminous plants. There are a good number of native leguminous plants over here, acacia and so on. So we tried to look for these, and we were successful in isolating a brady and a cynorhizobium. So that was a good breakthrough that we got in our research. And then, of course, I started working on other things, trying to explore, get a best thing. And that's where the other research started coming in on nanoparticles, red mud remediation, etc. Yeah, so that's kind of my journey to BITS Pilani. So should we start with the talk? I think I took a lot of time. I'll just share. Can I have the sharing access, please? Can I have the screen sharing access? Ma'am, you're on mute.
I need to unmute yourself.
Ma'am, at the bottom side, you can be able to.
No.
Share
I get a message that host has disabled screening share. So the host has to enable it.
Okay. Just a while, ma'am.
Yeah, sure. Okay. Yes. So is the screen visible?
Yes, it is visible.
Yeah, thank you, sir. Okay, so today I'm going to talk on synthesis of a novel biodegradable packaging material, which is embedded with metal nanoparticles. So here we are going to look at developing of packaging material, which is biodegradable, and we will also look at its application basically for packaging of fruits and vegetables, wherein these nanoparticles that are embedded are going to enhance the shelf life of the food. Okay, so before I begin, I'll just brief you about BITS Pilani. BITS Pilani is a deemed university, and it started in 1946-47 by the Birla family as a small engineering college. And before 46, it was a tiny pathshala in the village Pilani in Rajasthan. So in 46-47, we established the College of Engineering as well as the College of Arts and Pharmacy. In 1964, we acquired the deemed university status. Prior to that, we were under Delhi University. And from 1964, we have been independently working as a deemed university. We had the second campus of BITS Pilani come up into the year 2000, and that was the Dubai campus. So Dubai campus now is 22 years old and has been established understanding the need of the NRIs who have been living in the Gulf countries and would have to send their children back to India for higher studies. So this was established with the permission from UGC as well as the ministry in the Dubai to jointly have this engineering institute over there. In 2004, we had our third campus. There is a Goa campus established, and then in 2008, we had our fourth campus, that is Hyderabad campus, which added. And last year, we added our fifth campus, which is BITSOM in Mumbai, which is a school of management. So currently, four campuses, and the fifth one, four campuses which are solely engineering-based campuses, and the fifth campus, as I mentioned, in Mumbai, is basically for the management studies. Apart from this, we have a virtual campus, which is mainly for the work-integrated learning programs. So let me just start with today's talk. If we look at the current world population, we know that it is increasing day by day. And by 2050, we expect it to reach around 9.7 billion. And for this 9.7 billion, we always have this issue of feeding. So food and agricultural produce is a major concern that remains with all of us. This concern comes due to the limitations in the land that is available as well as the air water sources. And with increase in population, definitely there's this increased demand which we need to provide without harming the environment, because we are already seeing a lot of adverse effects happening on the environment due to unmonitored agricultural practices, like it could be addition of or an abuse of the chemical fertilizers or chemical pesticides, which are now accumulating in the environment and now getting into the food cycle. So bioaccumulation is, or biomagnification is something that we are really concerned of, and therefore we are moving now towards organic practices. But these organic practices have again a limitation. We can work to a certain extent, because what happens to the produce after it has been made? So to solve this, one is we can increase the agricultural produce, and there are a good number of people working on this particular area, trying to increase the agricultural produce by bringing in saline lands, desert lands under the cultivation practices. So that's one area of research. And the second area of research would be that whatever produce we have, we now prevent the spoilage of this produce. You must have heard, and we do read a lot in India about these things, wherein an unseasonal rain comes and washes off the entire crop. Yeah, so such post-harvest spoilage has to be prevented, and our work over here is a small attempt to try and find a solution for this post-harvest. So here we can look at some solutions which involve introducing inventive methodologies, definitely with eco-friendly solutions. We can also prevent the post-harvest losses by preventing or by improvising the storage as well as packaging conditions. Currently, if we see most of the agricultural produce in India, we don't even have storage rooms to put it. Yeah, they are stored on farms, they are stored in covered areas, and as I said, one rain can wash it off. So packaging materials to having proper storage environments is something that we need to look at. The currently used packaging material comes from recalcitrant petrochemical plastics, and these pose a huge hazard in the environment. And because of that, we are already struggling with the one-time use plastic or the low-grade plastics which are accumulating in the dumps. So to overcome all this, what we are proposing is to develop a biodegradable polymer, which will make the environment sustainable. It can save the environment, and at the same time, if we can make that polymer antimicrobial, definitely it can help in extending the shelf life of fruits and vegetables. So our aim here is to synthesize metal nanoparticles, and what we have done over here is aimed at looking for copper and zinc. We did not aim for silver because there's a lot of work already done on silver, and we wanted to establish the application of these copper and zinc nanoparticles. And the second aim would be to synthesize biodegradable polymers, which would be embedded with these metal nanoparticles. What is biotechnology, and what are these nanoparticles that we are talking about? So any material which has at least two dimensions, which range between 1 to 100 nanometers, would be classified as a nanoparticle. So nanoparticles, why are they so different, and why do they exhibit properties different, is mainly because of their size. So since the size is between 1 to 100 nanometers, their various other properties change. What we are seeing in this picture over here is basically gold, and this is how gold would appear. And if you look at gold at lower sizes, you can see that this metallic shining gold changes its color. So the properties like the surface property or the color, the absorption, everything changes depending on the size. So what nanotechnology does is we are looking at the same metal, but now its properties would be completely different, completely tangential to the normal metal or the metal salt because of the size. Metal nanoparticles can be synthesized using various methods. So we can make use of physical, chemical, or biological methods for making these nanoparticles. Under the physical methods, we could consider using ultrasonication, irradiation, microwave digestion, etc. In chemical methods, of course, we have the redox methods or the condensation methods or the salt gel methods. And under biological, we can use any of the biological extracts or any of the biological organisms, like you can use bacteria, fungi, plant extracts for synthesis of metal nanoparticles. So when we say biological, we are actually not using the bacterium, but it's mainly the enzymes from these extracts which are going to play the job. So if you look at the
projects, so like that, we have started working on PHB, ma'am, polyhydroxybutyrate. I've started isolating these granules from E. coli and Bacillus. We couldn't actually crystallize them and take them out in a proper way. We've just screened them with the staining, with the Sudan black bee staining, and we could actually confirm that they are PHB through the spectrophotometer. So I just had a doubt, how can we go further by crystallizing them and all that? Though I was reviewing the papers and all, I was not actually getting that protocol and how to go about, because since I've made few objectives of my research work, I'm
Madam, your
somewhere.
screen is visible.
Yes, ma'am.
Yeah, I just shared the screen.
Yes,
But
ma'am.
since Ms. Swati was talking, I thought we'll.
Yeah. So
Okay,
we were,
Swati, I'll
yes,
share
ma'am.
my
Yes,
contact
ma'am.
details. Yeah, and
Yes,
maybe
ma'am.
we can meet later sometime on a Google Meet or something, and I'll just guide you through.
Sure.
Yeah,
ma'am.
just before a speaker also talk, I wanted to tell Swati that we have actually worked on PSB from Bacillus cereus. She can refer our paper also. And you know
Yes,
that
sir,
E. coli
sure,
donor,
sir.
yes, this is Prakash Halami from CFT. Right? If you
Yes,
just
sir.
type it in Google
Yes,
also,
sir.
Halami, yes.
Yes, sir.
And
I
E.
So we have
just
coli
such
is
quoted
not
your
a good
words.
source. E.
Yes,
coli
sir.
is not a source for PHBs.
Yes.
Yes.
E.
E.
coli
coli
does
is used
not,
only for
unless
a
it's a
recombinant one.
recombinant E. coli.
Yeah,
It will
exactly,
not.
exactly.
So
Yes,
don't
sir.
try E. coli is what I wanted to
Yeah.
tell you.
Sure, ma'am.
It is a,
Yeah, Swati, we
it
can
is a
maybe.
yeah. Alkaligens latus is the one culture which produces maximum PSB.
Yes, yes,
Please,
sir.
madam.
Now let us go to the speaker, I believe.
Yeah.
So can we continue with the talk?
Yeah,
and,
yeah, sure, man. Otherwise, again, same problem will come.
Yeah. Okay. So I'll just make it very quick now. We'll keep our time limit on. Okay. So just a recap, we're talking about mechanisms of nanoparticle synthesis in bacteria. So bacteria basically use the bottom-up approach, wherein they start with the atomic from the atomic level or the molecular level. So what we can give the bacteria is a metal salt. Like for copper nanoparticles, you can give copper sulfate, zinc sulfate, and so on. And these salts definitely dissociate when they are in solution, and the ions are internalized by the cells. So that becomes the intracellular mechanism where the ions are taken up by the microbial cell, and then the enzymes, we basically look at the redox enzymes inside the microbial cell, which will reduce these ions into nanoparticles. And then we have the extracellular mechanisms as well. So in the intracellular mechanisms, as you can see, there is internalization of the ions. We have the enzymes acting on it. And then these reduced ions, that is what we have is the zero valent metal that is aggregated together to form the nanoparticles. In the extracellular mechanism, we have these metal ions in the environment, which would be reduced by the enzymes that are present in the periplasmic space, or many a times these are excreted out. So the secreted enzymes will convert these nanoparticles in the environment, convert the metal ions in the environment into the nanoparticles. So here we see external nanoparticle synthesis. Now, when we look at microbes, since bacteria offer both these mechanisms, what is useful for us definitely is to look for the second mechanism, that is the extracellular synthesis, because whenever it is intracellular, there is a limitation, because to recover those nanoparticles, we would have to lyse the cell. So that becomes an additional step for us, which could be time consuming, which could also require some amount of energy input. So because you'll have to lyse the cells out. So extracellular mechanisms are definitely beneficial and therefore less cumbersome, can be worked on easily, and recovery process, that is our downstream, becomes much easier. So how do we make these nanoparticles? It's extremely simple. So we take the bacterial culture, or we take the plant extract, whichever we feel is convenient, we want to use with. We add the metal salt and allow the bacteria to now act on those metal salts and start making nanoparticles. Now, in these synthesis of the nanoparticle is influenced by certain physical and chemical parameters. And based on the parameters that we use, the size of the nanoparticle can vary, as well as the shape of the nanoparticle can vary. So the different physical parameters could be temperature, aeration, which mainly influence a lot on the synthesis of nanoparticle, and chemical could be like pH, which also influences whether it is the acidic or alkaline range. So by modifying these process parameters, so we can always modulate the size as well as shape of the nanoparticles. So here you can see, by modifying the processes, we can get nanoparticles which could be in square shape, spherical shaped, triangular, hexagonal. You can make nanorods as well. So in our laboratory, we started working on arid soils, and from these arid soils, we hit upon a very good organism that was Ctenotrophomonas maltophilia. This was isolated from the garden soil, and we initially started working on this organism to look for metal resistance. And surprisingly, this was a multi-resistant organism. It exhibited drug resistance as well as resistance towards heavy metals. And then, because when we have to look for nanoparticle synthesis, that organism should be first resistant to the metals that we are trying to make the nanoparticles from. So looking at metal resistance is the first primary step, and we saw that this organism was resistant to more than 15 different metals. And then we realized it was a good hit, and we started exploring that if it is resistant, what are the different nanoparticles that it can produce. So when we studied that, we got a list of 13 different nanoparticles that this organism produced. Gold, silver, of course, is there. Apart from that, we had copper, zinc. The unusual, never reported before nanoparticles or the metal nanoparticles were also made by these, that is tungsten and molybdenum nanoparticles. So it was extremely good found that we got in this particular organism. So in this work, we are focusing on copper and zinc oxide nanoparticles that have been synthesized by the organism. So how do we go about the characterization to understand whether nanoparticles have been synthesized? One preliminary step could be to look at a UV visible, because generally if nanoparticles are formed, as I mentioned earlier, there is a change in the property, and this change in property can be also seen from the absorption of these nanoparticles in the UV range. So we can first look at the UV visible spectrum and look for a shift in the spectrum, which is a clear indication of the nanoparticle being synthesized. To confirm further, of course, a particle size analysis can be performed. So here you can see this particular graph. This is basically describing or showing us the size distribution. And we can see that the nanoparticles that are synthesized are mainly around 100 nanometers. And therefore, this was a preliminary work. And we realized that yes, 100 nanometers means it can be, there is a possibility of these being in the narrow range. We went on further after we got these nanoparticles as a preliminary confirmation. We went on to perform dialysis, purify these nanoparticles. And upon purification and dialysis, so we got another spectrum and understood that the size of the nanoparticle now was in the range of 70 to 80 nanometers. We did a scanning electron micrograph of these and saw a nice spherical unisized nanoparticles, as you can see in the picture. So these are the nanoparticles that we've got for copper oxide, and similarly, we did for the zinc oxide also, and for zinc oxide also, they were spherical nanoparticles that we got. Upon having isolated and characterized the nanoparticles, it was important to look at the antimicrobial properties. And these antimicrobial properties was tested against fungi. So we isolated fungi from spoiled fruits, and we got Aspergillus niger, Aspergillus cavirotus, and Penicillium chrysogenum. And on these three species, we tried to test the antifungal activity on a known concentration or known amount of spores. And you can see over here that the zone of inhibition is definitely going higher with the amount of nanoparticles that are being incorporated. Aspergillus niger was resistant to zinc oxide and exhibited no effect on, so zinc had no effect on this. However, it was sensitive to copper, whereas Aspergillus cavirotus and Penicillium chrysogenum both were sensitive to copper oxide as well as zinc oxide nanoparticles. So just to show you the effect and to show the zone of inhibition, it's a very clear zone of inhibition that we see over here for Penicillium chrysogenum. So these were the preliminary studies that we performed just to understand if these nanoparticles possessed any kind of antimicrobial activity. After having got these nanoparticles in hand, our second aim was to develop the polymer. And for this, we made a ternary blend of PVA, starch, and glycerol, where the polymer, these three solutions were blended to make the polymer. And into this blended polymer, we added copper oxide in one, zinc oxide in another, and in the third polymer, we added copper as well as zinc oxide nanoparticles. So these were the four films or the nanocomposite films that we made. One was with PVA, starch, and glycerol. Second was with PVA, starch, glycerol with copper, with zinc, and the fourth one was with copper and zinc both. These polymers we did an SEM of the polymers, and we also did the EDX analysis and EDX fluorescence analysis for these to confirm the presence of nanoparticles and absence of nanoparticles in the control film. So here you can see the SEM image of PSG film, wherein it's absolutely clear, a clean texture. And from the EDX spectra, we can see that the elements that can be found over here is only carbon and oxygen, and the fluorescence spectra of the same. That's the SEM image of PSG with copper. And you can see how that copper oxide has been embedded inside the film, and you can see that it's we're getting a granulated texture. So that smooth texture, what we saw of PSG, is gone, and you can see the clear embedding of copper. And this is reflected from the EDX and the EDX fluorescence that we did for this film. So in the EDX, we can see apart from copper, apart from carbon and oxygen, we also have copper that is being detected. That's the SEM of PSG with zinc, and similarly, we can see zinc being detected in EDX as well as in the EDX fluorescence. And that's the film that we developed for PSG, copper, and zinc, wherein now you can see in the EDX, copper and zinc both being detected and peaks for both coming up in the fluorescence, EDX fluorescence. After having made these polymers, it is just not enough to make the polymers and be happy. We need to look and establish the properties of these polymers to show that they are compatible with the regular plastics. Only then the applications would make sense. So here we did look at various properties. We tried to characterize these polymers by looking at the moisture content, by looking at the mechanical properties, and so on. So if we see, the first thing that we did was to understand the net moisture content of the polymer, and we can clearly see that the net moisture content of the polymer definitely with copper and zinc is less. And we require this less moisture content because that makes the polymer better. We want it to remain and be with lesser moisture so that it doesn't grab moisture from the atmosphere. The next one that we did was to look at the water solubility and the water absorption capacity of the polymer. So as we incorporated copper and zinc as compared to the control, you can very clearly see that, that the WA as well as WS, that is the water absorption capacity and the water solubility, both have decreased. That means by incorporation of the nanoparticles, what we can see is that the polymer does not absorb much water, which is again beneficial when we are looking at it as a packaging material. And similar water solubility has also decreased. That means the polymer is good and will not leach out into the solutions. We also check the hydrophobicity of the polymer by determining its water contact property. And you can see with the incorporation of copper and zinc, the hydrophobicity is increasing. That means this polymer is going to repel water and therefore can be considered as a good packaging material. If you look at the mechanical properties of this, we have compared it with PVA, and we can very clearly see that definitely the tensile strength is getting better. And if we see the elongation to break is also getting better with these polymers. So definitely a good mechanical property. That means we can say that our polymer is better, and though not as sturdy, we can say, as a PVA, but the flexibility is definitely better. It is getting slightly brittle because of the incorporation of the nanoparticles, but as compared to PSG, it is definitely getting a better result. Thermogravimetric analysis also were performed to understand the thermal stability of these polymers, because when we are using in packaging, these could be stored in any place, as well as understanding the temperatures that we have in Dubai over here. We wanted to look at the thermal stability of the polymers as well. And we can see that that also is good, and the incorporation of nanoparticles is not much affecting the thermal stability of the film. We studied the degradation properties of the polymers, and these were done in two different setups. One was wherein we took sterile soil and we cut the polymer into pieces and we put it into the sterile soil. And the second one was in non-sterile soil, just to see the effect of degradation. So definitely degradation was better in the non-sterile soil because it had microbes in it. And what you can see over here is a very clear degradation picture of these films. So from the graph also, we can clearly understand how the degradation has taken place. And if we see that the PSG film is definitely getting degraded better. But however, incorporation of nanoparticles slows down this degradation process, right? That means, which is also beneficial because we do not want the polymer that is being made to just degrade off. Yeah, we want it to remain for some long time. So it should have a good shelf life till its use is done, after which, of course, it can be degraded. So there is a good shelf life that we can see. It's not that it is a polymer which is biodegradable. That means it just vanishes in no time. So there, it is going to remain for a considerable time where it can be used and reused before its shelf life is over. Since the degradation was observed, we tried to look if the nanoparticles are being leached out from these polymers. And if we see the leaching of these nanoparticles, what you can see is over here, the concentration leaching of copper and zinc ions from the copper and zinc films. And here you can see the leaching of copper and zinc ions from copper zinc film, which is incorporated with copper and zinc both. And you can see that there is a small amount, it's not a large amount of leaching. There is a small amount of leaching which is happening, but that leaching is essential because unless these nanoparticles leach out, they will not exhibit that antimicrobial effect. We tested these polymers on strawberries, where you can see strawberries wrapped with the different polymers. And in this picture below, you can clearly see that the strawberry that was wrapped with PSG is completely spoiled after 7 days with a lot of fungus growing on it. Whereas the films that had incorporation of nanoparticles are intact. The strawberries still look good after 7 days, and this is at room temperature. Yeah, we know strawberries don't last at room temperature. They just get spoiled in no time. And that is why we selected strawberries for this work. And we have done a similar work on raspberries also, because both of these are highly prone to degradation and contamination. So this clearly indicates a good extended shelf life of strawberries due to these films. So coming to the conclusion part of it, we were capable of making these biodegradable polymers with PVA, starch, and glycerol blends. They exhibited good mechanical and thermal properties. So they exhibited antimicrobial properties and could enhance the shelf life of strawberries. There was no much leaching of the copper and zinc ions, and what we saw as leached is the bare minimum, which is essential for giving that antimicrobial effect. We did test these strawberries for their nutritional values, and there was no much change in the nutritional value. There was no transfer of these copper and zinc ions into the strawberries to so that that could harm the health-wise. So that did not happen as well. So there was no increase in the copper and zinc ion concentration of the strawberries. And therefore, it is absolutely safe material is what we try to claim. So this is the publication that we have got on this work, which is got published in April 2022 in Polymers. Extremely good journal. It has an impact factor of around 4.9. So that's for this particular topic. My team members are my students, and this work is being done with Dali Vilma Francis and Swarup Thalia Patil. These are both students. Dali is almost, she's a PhD student and almost submitting now. Saurabh is an undergrad student who very sincerely worked on this project on strawberries. I would like to thank our director, Dr. Srinivasan Matapusi, for his support, and of course, a big thank you to Shimadzu for helping us with the EDX fluorescence analysis. So some of the references, and I can be contacted on this email ID for any questions. Yes, thank you. Over to you, Dr. Pranita.
Yes, ma'am. Thank you so much.
I'm sorry, I just went fast because understanding the time limit that we have again.
So it would be
nice
really
presentation.
helpful. Yes, ma'am. It would be really very much helpful that you have covered your topic within that time duration, because within, I think, 5 to 7 minutes, this meeting will also be getting ended.
Yeah.
But yes, just Dr. Patinkar sir just had told that really your presentation was very good, ma'am, because your work is very great. By using that nanoparticles, you are using it as a packaging material. It is a very great work, ma'am. So
thank you, ma'am.
Yes, this recording we would circulate so that students will get benefit from that.
Yeah, sure.
Yes,
ma'am, I think Dr. Rahman has raised his hand.
Okay. So actually, I must be firstly formally thankful to you. Then question answer will be started. So because the meeting would be end after 5 to 7 minutes, ma'am. So on behalf of the organizing committee, Science College, Congress Nagar, Nagpur, and Microbiologists Society, India, we are very much grateful and very much thankful, ma'am, for sharing your views, sharing your information on this great work with our participants. It would be very much grateful, and because of your busy schedule also, you have given us the time for arranging this precious lecture from your side, ma'am. So thank you very much, ma'am.
Thank you so
And
much.
if the participants have any queries, now they can ask, or I think there are some questions in the chat box also, so that you can, there is one question, ma'am. How can we assure that nanoparticles are synthesized by the bacteria and there is no role of media used for bacterial growth?
Yeah, so we can always maintain a control. That is what we always do. I mean, no experiment is done without a control. So maintain a control with just adding the metal salt in the media. We have used LV media, so you can maintain a control, because you will need that control to initially see the UV spectrum. Yeah, so unless when we are looking at a shift only against the control. So a hypsochromic shift is something that we should observe in case there are any nanoparticles made.
Okay, there is one more question, that microplastic found in food chain, does this film would be vulnerable for this during degradation?
Basically, starch and glycerol would not contribute to anything, to getting into the food chain, because those are organic compounds. PVA also degrades fast. There are organisms which degrade PVA. It can go off completely oxidized into carbon dioxide water. So these would not enter the food chain, and that is why we selected these. They don't remain as the microplastic level.
Okay. There is one more, that the question, I think, yes. What could be the chemistry behind hydrophobicity of nanoparticles in the polymer system?
This is basically, we are looking at, when we are looking at hydrophobicity, it's the repulsion of water. When we are looking at contact angle, if it is done by adding a water droplet, and we look at the angle that is formed by the water droplet. Yeah, if it is not hydrophobic, then the water would be spreading on the polymer, so the angle is different. Whereas when the it is hydrophobic, the angle has a more steep. So we look at this hydrophobicity because in packaging, we essentially require the polymer to be hydrophobic. We do not want the polymer to, take up moisture, because that wherever there is moisture, that gives breeding to microorganisms. So the chemistry part of it comes because of the nanoparticles that we are using. Those nanoparticles contribute to this hydrophobicity, right? If we look at starch, definitely starch is going to bring in a lot of moisture. But if we look at these polymer, the metal nanoparticles, that is the quality of the metal nanoparticle that is making the film hydrophobic in nature. Yeah, so it's repelling water, because these bacterially synthesized metal nanoparticles have a protein layer around them. And that protein layer is to keep those nanoparticles in that zero valent form. Yeah, and that's indication. Unless there is a protein layer, we cannot say that the nanoparticle is biosynthesized. Then it becomes a chemical synthesized. So if you want to prove that the nanoparticle is biosynthesized, you have to show that there is a protein coat around it. Yeah, so there's always a layer around it. So all of these contribute towards the hydrophobicity. It's